So let's move on to the second session on animal models. As you will see, I'm not Jan Hero, and unfortunately, he cannot present today because he got a laryngitis and has no voice. And he has sent me a few of his slides last night. And because we wanted to have everyone know that we are working on a rat model for CLCN4. And so in this slide, you see the human CLCN4 gene shown. What?
If you go back to Zoom.
Ah, oh, sorry.
Is it right?
Oops. Yep. Hide this one. Thank you. So here we go. Human CLCN4 gene located on the short arm of human chromosome X. And as some of you know, at least 2 CLCN4 knockout mouse models have been published, although they don't show a phenotype. So they, at least the mouse model that has been established by the group from Thomas Jensch has been studied in very much detail, and there is no obvious phenotype. Why could this be? As we know is that in the mouse, CLCN4 is not on the X chromosome, but on chromosome 7. And there has other neighboring genes. And we thought this could perhaps be one reason why the mouse doesn't show a phenotype. And here you see the, here you see the sequence conservation between the mouse and human genes, this for CLCN4. But when you look at the rat, you see that there's much higher sequence conservation of the CLCN4 gene and also of the neighboring genes. And in the rat, CLCN4 maps to the X chromosome. Therefore, we thought, let's continue with the rats. And here you see that a comparison, sequence comparison on amino acids. The query is the sequence of the rat CLC4 protein, and the subject is the human CLC4 protein. And here is the alanine 555 valine variant, which we have talked about, and which showed a loss of function in the xenopus oocytes. And in the rat, in the human, it's at position 555, and in the rat, it corresponds to position 449. And also, as you can see here, is a very high conservation of the protein sequence in the mouse and in the rat, which suggests a similar function under strong selection during evolution. This slide is quite mechanistically, and what the team of Jan Hero has done is they have used the CRISPR-Cas9 system for introducing the missense variant right here into rat exon 11, and taking advantage of a repair mechanism which is called homology-directed repair, so that no nucleotides get lost. And this has been done by designing a single-strand oligonucleotide which has the point mutation. And there are a few other additional changes which will allow later on to genotype the the rats. And these are the three major steps in the process of engineering. First, the electroporation of the single strand oligonucleotide together with the Cas9 enzymes and the Cas9 complex into the fertilized oocytes of rats, and then implantation of the manipulated embryos in the rat mothers, and then afterwards screening of the F0 newborns at weaning, and this will at least take 6 months. Then in the second step, this will be the generation of the F1 carriers, because the F0 newborns are very often mosaic and will be crossed with wild type animals. And this will take at least 4 months. And in the next step, this is the confirmation of the mutant line, and which can be performed as soon as the F1 animals are identified and also shown to reproduce. That means that the mutation goes through the germline, which is also very important. And this takes at least 4 months. So this is a very busy slide, but what is important is that the number of oocytes which have been injected, or electroporated, is around 156. And from these re-implanted oocytes, they got 25 pups born, and it turned out that 9 of them carried the expected mutation. 6 still need to be confirmed, and this is how it looks. For example, the genotyping here, these bands on the agarose gel show wild type, represent wild type, and here you see at 3, which very likely have the missense mutation, and this has been confirmed then by Sanger sequencing. So at least one of the females has the missense variant at this position expected, and also one of the F0 males has this this single nucleotide exchange at this position. So I think we have been very lucky also. And here the chromatogram is simply bad. So where we are at the present, we are at we have several F1 rats. They are born and they will be genotyped in the coming weeks. So up to now, everything looks very good.